So I have spoken about MR-only planning for the past 10 years. So this time, I'm not going to talk about the MR-only work. I'll just give an update on 2 trials that we're running at the moment, but talk about some other work that we're doing related to MRI and radiation therapy, but not necessarily MR alone. Oops. So what I'll do, I'll just give an update on the trials that we're running at the moment. So there's 2 prospective clinical trials that we're running. One has just started. I'll also talk about a few different problems we've come across and possible solutions and work that we've done. So around automatic gold seed fiducial, intraprosthetic gold seed fiducial identification from MRI. Chat a bit about geometric distortion, how we measure that with MRI, and how confident we are with geometric distortion correction for MR-only planning. I'll speak a little bit about the rotating couch, particularly related to the MR-Linac project in Liverpool, and then speak about automated clinical trial QA, which is also linked to one of the prospective clinical trials that we're running at the moment. So firstly, the trials. So Hipster I presented a number of times here. So this is a phase 2 multi-centre prospective trial. So it involves Newcastle, the Calvary Mater Newcastle Hospital and Liverpool Hospital. So the aim is to test the feasibility of MR-only planning in a prospective setting. The idea was to treat 25 patients using this technology, and at this stage we've treated 15 at Newcastle. They finished their treatment, and we've had 7 patients complete their treatment at Liverpool, and we've got 3 current patients. So far all of the synthetic CTs generated from MRI have been clinically acceptable for treatment and used for treatment. So if you want to talk about it, come and talk to me afterwards if you don't know about these projects, because they're pretty exciting. So the really exciting news is yesterday we had the very last Hipster patient processed. So the MRI is the top row, the synthetic CT I generated last night. There's patients on the bottom, and I've just rendered the bone in the top right-hand corner of the screen, just to give you an idea. So that's cool. So these patients have the treatment, we'll write up that study, and so far it's all gone very well. The other study is the Ninja study, which is being led by Jared Martin and Mark Sidham in New South Wales. So this is a study where we've received Cancer Australia funding. It's a 3-year study looking at 2 sort of accelerated radiation therapy arms for prostate cancer. And apart from those 2 arms, there's also sub-studies for MR-only planning and also automatic clinical trial QA, which I'll talk about later on in the talk. The aim is for 150 patients to be treated, and that will be across 10 sites. So that'll involve an MR sim in each of those sites being installed, which is it's pretty ambitious as we only have 2 at the moment. So the first patient was scanned this week at the Calvary Mater Newcastle, and the first patient's consented at Liverpool. That was last week. So first patient scans come in, and that's going to be challenging. So this patient has a BMI of 40, which is the largest patient I've ever seen, actually. So we'll see how that goes. So I'll just go through a few previous studies that we've worked on, which might be of interest. So gold seed fiducials are used pretty much across Australia, as far as I know, intraprostatically. The idea is that you have these gold seeds, which you can identify on a digitally reconstructed radiograph from the CT, planning CT, and then for each treatment fraction you align those gold seeds with the portal imaging or cone beam CT that you acquire. If we're using an MR-only workflow, you don't have the gold seeds, so you can't align the portal imaging. So this is a problem. So what we were looking at for a while was the how we could automatically identify these and perhaps embed artificial gold seeds into the synthetic CTs for treatment planning. So this work was part of that. The first thing we had to look at was the quality of the MRIs that were being used for gold seed identification. So on the right-hand side is a T2 star-weighted MRI, and this is traditionally used for identifying these seeds. It's from a paper by Henk-Jan Huisman from Holland about 15 years ago, and it's fairly standard. But you can see the gold seed I don't have a pointer, but if I carry that little guy. This is a seed on MRI, so it's pretty hard to see. On CT that lights up like it's amazing. But you can see it's not a very consistent contrast. So what we've done is we've adjusted the flip angle of the MR. So we're using a T1 with an 80-degree flip angle to try and reduce artifacts from hemorrhaging and also to increase the contrast within the gold seed, which works fairly well, unless there's calcifications about the same size as the gold seed. It's not too bad. So we used that. And then we had a method where we generated an offline library of gold seeds based on a training database. So trying to identify what the gold seeds would look like in different orientations. So we used that offline atlas. We ran a clustering algorithm on those to identify the most common or the most representative orientations of the seeds that we'd need. Then when a new MRI was acquired of a patient, we'd run some image processing, so we'd identify the regions that were most likely to be gold seed fiducials, work out the size of those regions after we'd clean them up a little bit, and then we'd map that to the templates and get a sort of ranking of which locations within the MR were most likely to be gold seeds. And this worked fairly well. We validated that on 15 patients. We had manual contours from 2 experienced RTs, so we had an idea of inter-observer variability. We tried to get 3 and we couldn't for this study, but ideally you'd have 3 observers, not 2. And yeah, we reported an accuracy of 95%, but having said that, the expert RTs were only able to identify all 3 implanted seeds on 11 of the 15 scans, and this is a problem with using MR for the gold seed identification. We really need a better sequence or a better type of gold seed fiducial, to be honest. But yeah, the proposed method was pretty close to the manual experts. We were using this for the hipster trial, and we probably would have used it for the ninja trial, but we've since stopped using it because it's not reliable enough, to be honest. The problem's more on the physics, on the actual marker side, I think. What we did do is we had a method to insert the gold seeds back into the synthetic CT. So here's an earlier one that we generated, and you can see the 3 gold seeds on the DRR for this patient, which is pretty easy to do. But you want to make sure they're in the right spot. That's absolutely critical. Main problems that we have are calcifications, which looking almost exactly the same. Also, sometimes the gold seeds are implanted actually sort of right on the edge of the prostate capsule, so against the rectum, so they can be really hard to identify as well. So the third thing I wanted to talk about was geometric distortion. So we've done a few phantom studies, and I thought I'd mention this because it's come up in conversation and a few site visits. So at Newcastle, Peter Greer's group have developed a couple of, had a couple of phantoms built for their studies. So there's an anthropomorphic phantom which includes the rectum, bladder and prostate organs within the phantom, and there's also a grid-based phantom which we're using for validation. And really the aim of this is to look at the quality, to make sure the quality of the MRs that we're requiring match the quality of CT, and we're happy enough to treat patients based on those MR scans. There's vendor-supplied geometric distortion correction from Siemens, which we assessed. So there's a 2D distortion correction and a 3D distortion correction. And the basic idea is you take the phantom, you've got a ground truth from CT, you take the MRs and you map against the CT ground truth and just see where the distortions occurred. So with this study by Jidi Sun, one of our PhD students, he did that. He acquired the anthropomorphic phantom, did the CT scan of the phantom, mapped across the MR acquired images, and tested that with different levels of distortion. And so with the the distortion correction had a really positive effect. And this is why we're fairly confident with 3D distortion correction from Siemens. I think on this, yeah. So the maximum distortion across the pelvis was 7.5 millimetres. You can see from the image that as you go from isocenter out, the distortion increases, particularly with no correction. But with the 3D distortion correction turned on, we can restrict it to 1.7 millimetres. And within the organs of interest from the phantom, the distortion is less than 1 millimetre across those. So we're fairly happy with these results. This is a 3T scanner, yep. It would be interesting to try it in a 1.5 though. And in fact, we could just use the same phantoms. Yeah, that'd be another paper maybe. So there's another phantom which Amy Walker built in Liverpool, and this one's huge. So it's a sort of full size, it's meant to represent the field of view of a typical CT scan, so head and neck, say. And it's got these layers of fish oil capsules throughout. I think they're fish oil or vitamin E, one or the other. Anyway, she had to insert thousands of these little capsules into the phantom, which means we've got heaps of really good landmarks to assess geometric distortion from this. And the idea was to look at the 3T, at least the Skyra has a TIM-CT feature where the couch moves, and it lets you take fields of view from your scan which kind of match the CT field of view that you used to. That way you don't need to stitch multiple images together. Yeah, so this was 530 millimetres long. And so Amy followed the same methodology, so ground truth CT, acquire images, register the MRs back onto that CT and look at distortion, work out where the deformation occurred, do landmark target registration errors from the MR onto the CT. She's visualised the deformation fields using one of our tools, which is free to download, called Smiley. And yeah, she was able to publish those results. So she found that geometric distortion was less the slower the table went, which is pretty intuitive. She'd find that the best geometric accuracy was achieved with a table speed of 1.1 compared to 0, which was a bit unusual. I'm not sure why that is. So it's the middle image on the right-hand side that had the less geometric distortion. I'm not sure why that is. So, sort of related, I wanted to mention the rotating couch work. So conventionally, with a LINAC, of course, you have a rotating gantry around the patient, which is fine. If you wanted to use an alternate approach, a smaller system, more portable system, you could rotate the patient within a stationary beam. And this is pretty funky, but it's kind of the model which is being looked at for the MR LINAC in Liverpool. And also I've grabbed one of Brad Oborm's slides with proton therapy, where the same issue occurs. So the aim of this work was to design and test an MR-compatible patient rotation device for the MR LINAC. You could have the patient sitting in a chair that rotates around, but there's issues around, there's pros and cons. So we wanted to look at having a patient lying supine or prone. So here's Brendan Whelan, he's lying inside this device. And this couch design let us rotate the patient in, I think it was in 10-degree intervals around. So we did a study where we rotated volunteers around at sort of 45, 90, 135-degree angles. And then we took MR scans of the patients, those orientations, and looked at the amount of distortion caused by gravity for those patients. Yeah, with a view for the MR LINAC. And to do that, we acquired these images. The quality is not bad. We put him inside a Skyra. It's not actually the MR LINAC that we acquired these images from, but it's a sort of pre-MR LINAC study. So we did the rotation, acquired Brendan's images. We registered all of those back to a 0-degree ground truth sort of image that we acquired at the start, and then we mapped the deformation that's occurred during rotation against those scans. So we have a sort of inverted idea of what changes when, and the results weren't too bad. There's limitations to the study, of course, but as far as I know, this is the first study that's looked at this. So it showed that there's some, you could probably do a simple adaptive strategy where you rigidly shifted the beams to an automatically segmented region, so in this case a prostate. There's heaps of limitations to the study. So this was an N=1 study that we published. We've since got a PhD student, Jared Buckley, who's working on both female and male patients and rotating those. So that's volunteers and actual patients. So some of the limitations are around patient size. So it's a pretty narrow device. Then again, the gap for the MR linac is really narrow as well. So weight's an issue. The device still needs to be manually rotated, so it's really slow. I think with Brendan's scanning, it took 40 minutes, so you'll get bladder filling and so on during the acquisition. And also it's difficult to get in and out of the system as well. But it did enable us to quantify the amount of distortion and changes in organs during rotation, which was quite nice. The final thing I wanted to look at was automated clinical trial QA, and this is not just MRI, but we've been developing this on an MRI study, and it's applicable to either modality. So this is an NHMRC funded study with Lois Holloway from Liverpool. It's really important to have high quality contours for clinical trials because these form the basis of future decisions and results and guidelines. So what you really need to do during a clinical trial is to manually check every single contour that comes in for that trial. So have a PI or a group of PI or trial people checking those contours, rejecting them if they're no good, if they don't match the trial protocol, and also checking the planned dose as well, which can be affected by the contours as well. So this whole exercise can really add to the cost of trials and slow them down, and also affect results. In practice, what happens is a random sample of patients will be QA'd rather than every trial patient, as I understand. So yeah, the aim of the work that we're doing here is to develop an automated method of doing the clinical trial QA based on an atlas which has been developed at the start of the trial. So this is a pretty busy slide, but the idea is to, at the moment you'd currently get a benchmarking exercise at the start of most trials for contouring. The idea is to have that benchmarking exercise being done on a sort of a representative group of images, have multiple observers on each of those images, and then generate an atlas based on those. Ideally, it's not a single image, which is often the case at the moment, at least 5. And then you look at the tolerance criteria for those atlas-based contours, look at where they're acceptable, where they're not. And then you'd apply that to new images as they came in and compare the results against the trial contours that have been submitted. And the idea is to ensure that there's consistency across the trial data and also to improve the quality of the trial results. So yes, the aim is to use a gold standard set of contours and then come out with a report to flag, or like a traffic light report or something like that, to flag suspicious areas which can come up for a manual reviewer to check, to validate. So it's a clinical trial PI aid in a way. So there's 3 main approaches to doing it. There's an atlas-based approach which relies on image registration, so it's slow. The idea is to include inter-observer variability into a probabilistic atlas, map that across, and that gives you information about local error within the contour. So you can pinpoint those areas where a clinician's made they're outside the usual expected variability for a contour and highlight that region. There's a classification-based approach which is super fast. This is in a matter of seconds or so. based on radiomic features, so we can quickly identify shape-based features, texture-based features that don't match the atlas set, which is suspicious as well. And then, of course, this is basic structure-based features, so volume, curvature, and things like that. For example, looking at the volume of a structure compared to the expected population norm for that structure. So the software has really got a couple of inputs: the clinical trial atlas set and your clinical trial cases. The outputs a report with visualization of areas of concern and some sort of ranking. We haven't actually finalized the report, and this is still work in progress that I'm showing. But in terms of software, we've tried to make it cross-platform, so we can use it both across any operating system. But we're interested in using it in a distributed learning setting, where we go back and look at retrospective data and look at whether that meets compliance with protocols and highlight differences and so on. We need it to be accurate. It needs to be robust, so particularly across different field of views, which we've come across already. Also image artifacts. This is more of an issue for MRI-based analysis. Of course, most of it will be CT-based, where there'll be artifacts and so on that we need to contend with. It'll need to be scalable, it needs to be extensible, so from small trials to mining large data repositories. The common thing is that we'll never have very many contoured atlas images because they just take too long to get. So we'll always be working with a really tiny atlas set, which is interesting compared to, say, a deep learning approach where you have a lot of training data. So to validate it, we've been involved with the Prometheus study. This is a phase 2 multi-center study looking at stereotactic radiotherapy. It's similar to the Ninja, to be honest. So Ninja's kind of Prometheus Mark II. And part of that study was there was an MR component where men had they had spacer gels. This is a gel which is injected between the rectum and prostate to try and move the rectum away from dose, reduce toxicity, and also a rectifix device, which is kind of like a cricket stump, which is inserted into the rectum to hold the rectum straight and away from the prostate that's been treated. There were 5 observers in 4 different centres across Australia, including in Brisbane at the PA, and we had observer contours for the prostate, rectum, bladder, and also the spacer gel and the rectifix. So here's the result of the 5 observers on 2 of the scans. So this is actually pretty good for inter-observer variability from what I've seen. So the rectifix is that round area in the rectum, prostate, space, bladder. On the right-hand side, you've got the other organs. So interestingly, there is an outlier with the prostate segmentation on the right-hand one, but this is quite a lot lower. The superior. So from these scans, we can generate average atlases. Now these are, to do this, we take out one exemplar sort of case, which represents the population. We've only got 5 cases, so we've taken the average size prostate, I think, for this atlas set, and then you register the other cases against that exemplar image, and then you keep iterating against that average image, usually sort of 3 to 5 times to generate an average atlas. And then you use those same transform deformation fields to map across the manual contours. So you get these average representations of the organs across the atlas set. So these would usually be smoothed a bit, but I've just left them in their natural state. But you can see the rectifix quite clearly in the other organs. So now that we've got an average representation of these organs, we can map that across to new cases and look at whether they meet the trial protocol. And of course, these are probabilistic as well. So we have an idea of how the inter-observer variability mapped into that atlas space. In this case, there was one bladder which was really, really small compared to the other 4, which is why it's not 100%, or probably one in the middle here. Otherwise, it's converged quite well, average atlas. In terms of classification, we originally used texture features from the whole organ. We realised that what's important is the boundary around the organs in terms of texture. That contains the most important information to help the classification results. So we typically just do a sort of a edge region around the organ boundaries. And then we augment some data. We had, for Prometheus, we did have some fail cases and pass cases, so cases that were corrected by a radiation therapist as part of the trial. So we had some cases that were incorrect. We used PyRadiomics, which is an awesome Python package for extracting features. We used a random forest classifier. We don't have a lot of data to play around with, so this worked quite well, and it's extremely fast. And we use that to identify cases that might be pass or fail. So here's an example of the rectifix, where the green contour doesn't meet trial protocol, the blue one does. This was correctly identified, basically because of its shape. It didn't match because we're looking at features relating to curvature or flatness or sphericity of the contours, then this the bumpiness came up as an unusual feature of that contour. So with the atlas, we can use that atlas set. We have an atlas image. We register that against the contour or the image that's been tested in the trial, and we propagate across the prostate or organ contour. And so we have an idea of how well the observer contour matches the track or the atlas contour, which is in green. The original contour is in blue. So in this case, it's a really good agreement. Dice of 0.89, which is really good, and the volumes are quite similar. What we can also do, because we have probabilities on the atlas, we can look at the 30% agreement, 70% agreement. We can do any percentage of agreement and look at where the contours that the experts contoured on the image, and look at how well it fits into the range of values that are expected. So for the prostate, you'd expect the base and apex to have some greater intra-observer variability rather than other locations. And in this case, this contour is well within the boundaries you'd expect. So it's just a way of highlighting where the contour might need to be corrected. We also have probability, so you can directly map the probability without thresholding it as well to highlight different regions of differences. Probably make it a bit more attractive than that slide though, to be honest. So this is my final slide, I think, but for the Ninja study, because we've got an automated QA component to that study, we'll be moving from the Prometheus developed tool. We'll be adapting that to the Ninja study and validating that in an initial subset of manually reviewed Ninja data. So it's 4 patients per site. So we've started, we'll start doing that very soon for Newcastle. After that, we'll move towards using it in real time. So the ultimate dream would be to have this something that could be used for retrospective data, but also something that could be used in real time. And even as somebody's contouring, to highlight errors or highlight regions that don't match the clinical protocol in real time as they contour an organ, which would be that would be quite cool. But yeah, for this study, we'll be flagging cases that need manual review and returning that back to the PIs. Yeah, that's my final slide. So yeah, I'd like to acknowledge cast of thousands who've helped with these projects, particularly Lars Holloway and Peter Greer, and funding support as well. So thank you.